Hi guys, welcome to my discussion 7 video. Today I'm going to be going over mechanisms of eukaryotic gene regulation, particularly focusing on acetylation and methylation. You should remember that last week we talked about prokaryotic gene regulation, and that was with the operons, where we had multiple genes that were being controlled by a single promoter and operator sequence. With eukaryotes, it's a little bit different, and there are more mechanisms than just acetylation and methylation, but these are just the primary ones that I wanted to focus on today. So firstly, talking about acetylation, all that we are talking about when we're referring to acetylation is the addition of an acetyl group to the lysines on histone proteins. So you can remember that in eukaryotic cells, the DNA is going to be wrapped around these histone proteins in a structure that looks like this. So these little pink things are going to be the histones, whereas the blue strands, the actual DNA itself. And so why this is important is that histones are generally going to have a positive charge when they're not acetylated. And this is going to cause the histones to really want to interact strongly with the negatively charged phosphates on the DNA. So if we look here, this real tight packing is occurring because the histones want to maximize the amount of interaction that they're having with those negatively charged phosphates. So how acetylation works is that it essentially just neutralizes that positive charge on the histone. And then that causes those interactions between the histones and the phosphate groups to kind of fall apart. So then we can kind of relax into this more loose structure because those interactions aren't going to be pulling them tight together anymore. This is a reversible process. So we have different enzymes that can cause the acetylation as well as the deacetylation. So once we acetylate, it's not like it's forever going to be that way. And that's kind of why this is a really important mechanism in terms of gene regulation, because we can have genes be transcribed under situations where we want them to be transcribed, but then when there's situations where we don't want them to be transcribed, we can temporarily turn that off, thus saving the cell energy and resources and stuff to focus on things that it actually needs at that moment. So again, here's just a slide that shows the different types of enzymes that you can use to achieve this. So HAT, which stands for histone acetyltransferases, that's going to be the thing that adds the acetyl to the lysine, which is going to cause this structure to open up. And then HDAC, histone deacetylases, is just going to reverse that process, remove that acetyl group. That charge, that positive charge, is going to be restored, and then the histones are going to want to interact with that phosphate group again, causing this structure to reform. So the role of acetylation in cell differentiation and regulation is super important. Sorry, the slide is kind of wordy, but essentially all that it's really saying is that what it requires is just one single, what's called a pioneer regulatory transcription factor. A pioneer regulatory transcription factor is important because it's able to initially bind to this supercoiled structure. And that binding is going to recruit the HAT enzyme, which will allow for this to happen. So if you can think about this process, this wouldn't just happen naturally. We need to have that enzyme. And the enzyme isn't going to work on it unless it receives that signal to kind of come over and to act on it. And that is going to be in response to that pioneer transcription factor. And so hopefully you can remember from the lecture that we said that general transcription factors are going to be really similar across different cell types. So those ones are going to be relatively stable, depending, like, no matter what type of cell that you're looking at. However, the regulatory transcription factors are going to be really, really different depending on what type of cell, what situation that you're in. And that's really helpful because those regulatory transcription factors are going to bind to really specific places on the chromatin. So for example, if we wanted a gene to be expressed that was in this area, we would recruit to that specific binding site and allow just this portion to open. Because remember, the whole point of just relaxing a certain component is to just select the areas that we want to transcribe. So those regulatory transcription factors are going to be really, really specific in terms of what portion of the genome it's opening and allowing to occur. I think that's all that I really wanted to say. Oh yeah, I did want to talk about a little bit about enhancers and silencers, because I think some people get confused by thinking that those are what the transcription factors are themselves, but they're not. So enhancers and silencers are literally just DNA sequences, so they're just parts of the DNA itself, but they act as the sites that are going to cause those regulatory transcription factors to bind to them. And then once those transcription factors are bound, then that's what's going to cause that conformational change, which will either enhance or repress expression, which is why we have enhancers and silencers. They're both just DNA sequences, but we call them different terms just because they have different effects on transcription. Okay, so we can try to think, is acetylation a relatively short-term or long-term modification? And hopefully you can identify that this will be a short-term modification. And you could see that because we saw that the process was really easily reversible. We have these enzymes that can add the acetyl group, and then just as easily we can take it away and we don't need it anymore. The modification is also happening to the histones, the proteins, not the DNA sequence itself. So you can think when the DNA is being replicated or when we're reproducing, those histone changes that have occurred in the past are not going to affect what's happening in the future because it's not the actual sequence itself that's been changed. Okay, so now moving on to methylation. Methylation is going to be more of a long-term modification, and we can see how this works in just a second. Essentially, methylation is referring to the addition of a methyl group to cytosines in the DNA itself, so actually inside the DNA strand. This is carried out by an enzyme called DNMT, which stands for DNA methyltransferase enzyme. And essentially what it does is, if this is the picture of our strand that we're caring about, and say that these are the methyl groups that we've added, so here was the unmethylated DNA, and here's the methylated DNA. This can work in one of two ways. It can either just prevent the transcription factors that would normally bind here, like if there were regulatory transcription factors that could bind in this major groove, when those are added, now they will no longer be able to bind there. Or it can also work by having these methyl groups attract HDACs. So remember that HDACs were from the acetylation component, where we caused that supercoiling. We induced that supercoiling by recruiting the HDAC enzymes. And so the methylation can kind of be tied to acetylation by this signaling, hey, we want this, the HDAC enzymes to come over here, and then we can condense. Essentially, you're turning that portion of expression off. Okay, and we can see that more clearly with this picture here. Here is our unmethylated strand. And then we have this DNMT enzyme that's being added and adding those methyl groups to the cytosines. And what this will do is this will change the shape of the major groove. So the binding domain is going to change because of these additional methyls. And then that will attract those HDAC proteins, which will ultimately cause this to recondense. So we can think of it as being, we can think of it in terms of the DNMT enzyme will change the major groove, which will change the recruitment, which will change the shape. So that's kind of how it works. Methylation sites are often clustered near or within the promoter itself, which hopefully makes intuitive sense because we really want to be specific with what genes we want to either be expressed or not be expressed. So when we have those methylation sites at or really close to the promoter, we're able to be specific and saying, this is the gene that we want to turn off right now. If they were kind of just like randomly distributed throughout the genome, you would have situations where if you're trying to turn off one thing, you might actually be turning off two things that you didn't necessarily want to happen. You'll just get like a lot more unspecific regulation, which is not good. So that's why we'll generally see these sites being clustered near the promoter. And the sites that methylation most often occurs in are these like C and G linkages. And when you have a lot of them in a row, those are called CPG islands, which is essentially just this type of thing where you have a bunch of the C and G linkages all together, so that when that region is highly methylated, then it's going to be really specific and saying like, this is the part where we're super condensing the chromatin. And so this is just a picture just basically showing what those CPG islands look like. So we can see that it's inside the promoter, and we can see what it looks like when it's unmethylated versus when it's methylated. And then this is just demonstrating where we're adding the methyl group to the cytosines. Cool, so we can think, is methylation a relatively short-term or long-term modification? So hopefully you should be able to say that this is going to be a long-term modification. So firstly, it's basically an irreversible process. We know that we have this DNMT enzyme that will cause the methylation to occur, but there's really no known mechanism that we know that can undo this. So once you're methylated, there's really no going back. And since we remember that this methylation causes that supercoiling to happen, it's essentially silencing a gene. And since we can't necessarily reverse it, once that gene's off, we can't restore its function anymore. We don't have a way to control that. And also, it's a long-term change because the changes are heritable. Remember that we're changing the actual sequence of the DNA. We're no longer just making changes to the proteins that it's wrapped around. So now when we have DNA replication or reproduction or whatever, we're going to be passing along that methylation pattern, which will affect, obviously, gene expression in the new organism. Okay, so we know that methylation plays a huge pattern in eukaryotic cell differentiation. We just covered that. So we can kind of use that to predict what would happen under certain circumstances, what types of methylation patterns would occur. So I'm going to kind of go through each of these three, and I want you guys to think or like brainstorm what would happen with the methylation patterns for each of these. And then the next slide, I'll go over the answers. But so firstly, we have the situation where there's a region that codes for a gene that a particular cell type would never need. So we can think, for example, gene sequences that code for photoreceptor proteins in a kidney cell. We would never need that because the kidney cells are inside of our body, so there's really no reason that it should be able to take a light stimulus. There's no information we could really gain from that. So what would we expect for methylation to happen here? So try to brainstorm about that. Second situation, what if we had a region that codes for a gene that a particular cell type may need, but only in certain circumstances or under certain conditions? So for example, oncogenes that will induce apoptosis, we obviously really want those. We want those to be able to be transcribed if necessary, because that will play a huge role in cancer suppression and preventing tumors. But we don't necessarily want them always transcribed. We only really want those active when the cell is able to detect, oh, there's a lot of other cells of the same type around me, to the point where I would actually be doing more harm than good, so I'm just going to self-destruct. And then hopefully that will prevent tumors from growing. So we want those to be available, but not necessarily always. So try to predict what would happen in terms of methylation there. And then thirdly, let's say that we had a region that codes for a gene that a particular cell type would always need in order to function correctly. So we can think, for example, here, the enzymes in a liver that are responsible for breaking down toxic substances into harmless byproducts that can just be excreted from your body. That's going to be really, really important, because that's essentially the function of our liver, is to help detoxify our body. So if we knock out these liver enzymes, then essentially you're knocking out the whole point of your liver. So try to predict what we would expect for the methylation patterns here. All right, so hopefully you guys were able to get the right answers here. So in that first situation where we had the region that codes for a gene that a particular cell type would never need, we would expect that DNA portion to be methylated and closed. And remember that methylation is a permanent change, and it's causing that gene to be silenced, which is fine, because if we never need that type of gene, then we really don't want this to be reversible. We don't want a situation to accidentally induce transcription of this, because that would never be useful for us. So having it permanently closed is just going to make sure that we aren't expressing things that we would never ever need. Now, in the second situation where we have the genes, the oncogenes that we would sometimes want, but only in particular cases, we would expect that to be unmethylated and probably deacetylated until we have a situation that's going to induce that acetylation. So we remember that if it's unmethylated, it's technically going to be able to change. The structure can open up or loosen, so transcription can be blocked or can occur, depending on whether or not this acetylation or deacetylation is happening. So when it's deacetylated, it's going to be condensed and won't be transcribed, which is probably going to be the default state of these oncogenes. But then if a situation occurs where the cells detect we are getting way too much of this type in this one area, then this acetylation could be triggered, which would cause the transcription of those oncogenes. So that self-destruction could happen. So that apoptosis would happen. And thirdly, the region that codes for a gene that a particular cell type would always need, expect that to be unmethylated and open. Hopefully this is clear. We want this unmethylated, obviously, because we do not want those genes to be silenced at all. We really, really need those genes. And then we want them open or acetylated, so that that transcription can always be happening, because we're always going to be requiring those, no matter what the circumstance, really. All right, so that's actually all that I have to cover in terms of today's content. I did want to do a quick note about the discussion exercise. So on the very last page of the exercise, I believe it's slide 4, Problem number 2, parts C and D. It's the parts about beta thalassemia and 5-azacitidine. These are optional. At least I'm making them optional. I highly recommend at least looking at them and brainstorming possible ideas given what you know, given from the lecture and from today's video and from the exercise itself. I think it's a really good kind of application question, seeing how this material can actually be applied to like a relevant real world example. But I also don't want you guys to stress out so much about getting the exact right answer that you are wasting time that could be better spent like studying for the exam itself. So don't stress too much about it. I would recommend at least just trying, definitely reading it and just seeing what pops in your head, especially if you're working with a group. It's just like a really good way to kind of apply this material a little bit more. All right, thank you guys for listening and good luck studying for the exam. Thanks.